To the last talk of the session by Professor Birgitta Whaley. She's a professor at the University of California at Berkeley and director of the Quantum Information Center. She has many awards and contributed to important work both on quantum information in general, but also on kind of quantum information and life sciences. Maybe one very quick story, and then we can get started. Birgitta was actually one of the very early people to encourage me to start this work on protein qubits. So thank you very much for that. Okay, let's get started.
You're welcome. New insights and new tools. Actually, what I'm going to talk about today involves new experimental tools, certainly at the end. They're always welcome, and certainly new people to the field, no matter where you come from. So I'm going to talk about probing biosystems of single photons. And I have to say from the outset, I'm not going to talk about human health or anything medical. I'm going to talk about photosynthesis, which is a sort of another big area in quantum biology. And also, the talk will not be theoretical. It will be actually experimental. It'll be about work done together with my colleague Graham Fleming, who has done for many years leading work in photosynthesis. And two postdocs, Whaley Lee and Rob Cook and Li Wen Ko. And there is an experimental postdoc position available for these quantum light experiments. I'll say more about that at the end. So the motivation is to understand the initial stages of photosynthesis. And this has been a topic of experimental and theoretical work for probably more than 30 years, probably even up to 50 years, using lasers. But lasers are in the first era of quantum biology, which is prior to 1960. Prior to, well, the lasers actually introduced, I would say, the second era, which is started in 1960. But there's a lot of open questions, which I'll just touch on peripherally, about how much you learn from an experiment where you're exciting system with higher intensity light than you would do in a natural system. So the lasers used typically require, you have to actually excite about 10% of the chlorophylls in the system, and that's corresponds to a very high light intensity. So what we're interested in is understanding how the light comes in, it's absorbed somewhere on the antenna of a light harvesting system, and then there's this excitonic, and now it's understood to be also vibronic, process of transfer of energy to the reaction center, where electron hole pair separation occurs, and the electrons go on to do the chemistry and to store energy in chemical substances. And this first part of the photosynthesis has been possibly the most actively studied over the last 30 years using spectroscopy tools. And so it's very important to understand, because 50% of green matter on Earth does actually use this particular complex. But I'm going to talk about a somewhat simpler complex. And before I do that, let me just say one word about other things you can do with single photons. To our knowledge, the experiments that I will present to you are the first experiments done with quantum light on a photosynthetic system. By quantum light, I mean controlled single photons or pairs of photons. But there have been very interesting experiments done by Krivitsky and his group probing vision with single photons. And this is also a topic of considerable history in biology, or in the interface in biophysics, this one primarily. People have been looking for, again, also about 30 or 40 years at the ultimate limit of sensitivity of the eye of humans. And what Krivitsky's group did was they looked at, not humans, but they looked at this cute little toad here, Xenopus levi, and they carried out experiments with single photon sources. And this is a really cool experiment because they could actually, so they're carrying out experiments on an entire rod cell in the eye, and they're looking at the response of the rod cell to triggers by single photons. So it's a big step down from looking at humans looking, recognizing single photons, which is a good thing in terms of understanding. And then they could, because they're looking at an entire rod cell, they could then correlate, they could then measure the electrical signals coming out from the cell. So they correlated the input photons with electrical signals coming out. And that's really where we would like to be eventually with our photosynthetic studies, namely to correlate the single photons going in with the electron hole pairs being produced. But right now the technology, it's too much technology to put together. So we're going to look at just the correlations between the input photons and fluorescent photons. And so that'll be the next slide, I think. So this one here is just to, maybe for those of you that don't know much about this area, just a sort of a summary slide for the kinds of studies that I think someone also mentioned. There's lots of work done with lasers on photosynthesis, and that's the earlier work, which has really produced a huge amount of very valuable and useful insights. And it's going down to femtoseconds. But again, but using ensemble types of measurements. So the motivation for our going down away from lasers and going down to single photons was consideration of the meaning of quantum efficiency. So quantum efficiency in the context of photosynthesis means that one photon from sunlight is transduced to one electron hole pair. So this is typically measured with something like this little device here, which you can actually go and buy. It's actually a quantum meter. Well, before people started talking about this kinds of analysis in terms of actual quantum light. But it's usually measured, it's measured by fluorescence counting. And so you can see the expression here in terms of rate constants, and the rate constants are measured either in the dark, when the plants or the bacteria are not illuminated by light, or whether it's fluorescence under light conditions, and there's some minimal light condition to define the dark. So this is very much a bulk measurement. You're averaging over reaction rates. And the question that we raised was, well, what is the mechanism and even the timescale for the absorption of one photon energy equivalent from sunlight? Because if we need to have one photon transduced to one electron hole pair, we need to have energy to do this, to release the electron pair. And surprisingly, when I first started to think about this some years ago, we looked around in the literature, there really wasn't even any estimate of the timescale for the absorption of one photon. So I'm not going to talk about the timescale today, but I will talk about the efficiency. So this is a summary slide about sunlight. Sunlight, left is filtered blackbody radiation, has ultra weak intensity, less than one photon per mode, much less than one photon per mode. The flux, if you want to keep a number in your head while I'm speaking, the flux is on the order of between 100 and 1,000 photons per nanometer squared per second. A second is a very, very long time in the biology, in this kind of, in photosynthetic times. And there are very weak coupling to chromophores. The coupling of the light to the chromophores is much weaker. In fact, this was also what surprised us. It's much weaker than the other famous energy scales of photosynthesis, which are the the exciton-exciton interaction energies, the exciton-phonon energies, and the phonon-phonon energies. Those are all roughly similar, and there's good reasons for that, or at least we believe there is. But this coupling to chromophores is orders of magnitude less than that. So getting the light into the system is a real bottleneck, and it's a bottleneck that the photosynthetic systems have to deal with, and for more about that later. So what we are interested in, okay, and then here's another perspective about light harvesting. That you can think, if you're a physicist, you can maybe think of the light harvesting system as a photon detector with a very high internal quantum efficiency. That's the engineer's perspective. This is an internal quantum efficiency because it's the efficiency of the absorbed photons being transduced into electron hole pairs. And a big question in this field is whether coherence is important in these processes. So if we're thinking about sunlight, our experiments will not be done with sunlight, but let's just think in terms of motivation. Our experiments will be done with coherent photons, coherent single photons. So sunlight is often described as incoherent. And if you take it as an ensemble, it is indeed very incoherent. But just how incoherent is it? Well, you can estimate the coherence area and also the coherence length.
Sunlight is program. It's great to have them come to SLAC to bring their energy and enthusiasm to the laboratory.
Okay, I don't have Wi-Fi. I switched my Wi-Fi off, so I don't know where that's coming from. But anyway, it is indeed great to be at SLAC.
They're just
It's from Lynn. Oh, maybe he could mute. Yeah. Okay. So anyway, so the bottom line here is just if you take these numbers, which are well known in the literature, and you multiply them together, you get a coherence volume, which is something that Wolf talked about a long time ago. And this coherent volume is about 53,000 micrometers cubed. And that's significantly larger than the volume of chloroplast. So as far as a green plant is concerned, or as far as a particular chloroplast and set of chlorophylls are concerned, a photon from sunlight would indeed be coherent. And there's a very beautiful experiment, I won't go into the details of this, from Zhanwei Pan's group in China, showing that if you have the right detector, so in other words, if you, in their case, if you polarize and spectrally filter sunlight, have fast temporal detection, and you can then as a proxy for the electron hole pair creation. So this experiment actually doesn't require any entanglement. We will only need to have correlations between two photons, which are produced from spontaneous parametric down conversion here. And so then they are coming out here, and one goes into the sample, and fluorescence is measured from the sample in this detector, and the other one goes into another detector, counted, and acts as a herald for the fact that a second photon was incident upon the sample. So the actual experiment is done not on the green or not on the PS2, but it's on the simpler system, which is known as Light Harvesting System 2, LH2, which is the outer antenna in the purple bacteria. And this looks, certainly to physicists, this looks much more amenable. It looks like it has some degree of symmetry, and it does have some partial symmetry here. It consists of two rings of chlorophylls. One ring absorbs in the blue and at the 800 nanometers, that's here, and that's the blue ones indicated here. And the other ring absorbs in the red at 850, or to the red of the 800 nanometers at 850 nanometers, and those are the red ones here. And the way that in nature this works is that primarily the B800 ring absorbs light, and then there is some energy transfer from the B800 ring to, in this picture it would be down, to the B850 ring. And then you see on the top right a TEM of a sample of these in vivo. You see the small rings are these LH2 rings, and the excitation will then jump from one of, in practice, the excitation will then jump between these rings and eventually get to a big ring, which is known as LH1, which is where the electron hole pair separation occurs. So we're going to take the fluorescence in this. So this experiment is carried out with just a set of LH2 rings, no LH1, so it's not an in vivo experiment. It's done on a sample in a gel of extracted LH2. And we're going to take this fluorescence from the LH2 rings as a proxy for the eventual electron hole pair separation, because it's the first step along that way. So, and it's the simplest experiment one can do, starting out with this first quantum light experiment. So what we do is we measure, we have our photon pairs produced by the spontaneous parametric down conversion here. They're produced at 800 nanometers. One goes towards detector 1, which just tells us that yes, a photon pair was emitted. At the same time, the other one is incident then on the ring. So we count all of the Herald's photons at detector 1, and then we count all of the fluorescent photons coming from the B850 ring, and everything's filtered, so we only have we don't have any 800 coming into detector 2. We count the fluorescent photons. And so just first of all we first check the coherence, and we make standard coherence G2 measurements on these photons. So for the incident light, that is so it's basically it's gated on the Herald, and then we get a very low. Which one is the incident light? The blue one, yeah. The blue one, we have a very low incident light, and it's basically 4 standard deviation below the cutoff for single photons. And so this definitely is a definitive demonstration that we have indeed single photons incident upon our LH2 ring. And then for the fluorescent light, we then measure, again here, if we have single photons incident, we can then still use gating to measure then the G2 of the fluorescent light. And so this is for the heralded photons. And so the error bars are somewhat larger here, but we conclude that we can conclude that we have single photons for both incident and for fluorescent light in this experiment. So this experiment was entirely done with single photons. And here's the input that I gave from quantum information is I suggested that we look to check, we look to see the meaning. Actually, it wasn't really inside of quantum information, it's more from molecular dynamics. I was suggesting that we check this quantum efficiency, whether it really has the same value and the same meaning at the level of individual photons. So this is the analog of studying molecular reaction dynamics people did for many, many years before the 1980s in bulk and extracting reaction rates, and then going and doing molecular beam experiments where you take individual molecules and collide them together and look at the dynamics. This is the analog of that for the photosynthetic system. So what we do here is we take all our counts of Herald and fluorescent times, and then we construct a histogram of the number of Herald photons detected per fluorescent photon. So here, for instance, so if everything's perfect here, we have perfect efficiency of fluorescence, then you would have, for each Herald, you would have 1 fluorescent photon. But actually the fluorescence efficiency is more like 10 to the minus 4, which starts to make you realize these are hard experiments. And then you'll have many missing fluorescent photons. And you might have here, for instance, 3 Herald photons between these 2 fluorescent photons. So then you have to put something here in number 3. So we bin these, and then we look at the distribution shown on the right, and we do some simple theory, relatively simple theory, and we can show that there's a geometric distribution for the probability of this number of Heralds per fluorescence. In other words, how many photons are absorbed in order to get a fluorescent photon out as a proxy for electron hole pair separation. And if this efficiency is very, very small, then this becomes an exponential. So what we find is over here, we find that indeed the maximum probability is n is 1. So the conclusion is that 1 fluorescent photon requires just 1 absorbed photon. So this is the microscopic demonstration of the quantum efficiency at the single photon level. And it agrees with the bulk what the conclusions from the bulk studies, although the bulk studies never quite get to 1. There is a small amount here at 0, and this comes from 2 fluorescent coincident counts, and it's actually quantitatively consistent with our G2 measurements, so there's not an issue there. So that's the conclusion from this experiment, that the efficiency is near unity, or is unity in ultra-weak light, even at the level of single photons. So the second experiment, that one was published, this one is not yet published. Second experiment is, what is the role of photon statistics in photosynthesis? This is again a topic that people in photosynthesis and people who think about quantum biology and photosynthesis argue about a lot. There are people who say that any laser study is completely irrelevant because it's not incoherent sunlight. And so you should have thermal light statistics. So I've summarized here the statistics of single photons and the probability and the statistics of thermal light. They're very different. So our goal here was to see, well, if we measure the statistics before and after of the incident light and the fluorescent light, is there a difference? Has the molecular system, has the complex, the biological dynamical processes of energy transfer going on in this complex, does it change the statistics of the fluorescent light? And so here's the second experiment where now we consider, we're comparing the single photon setup that I showed you before with a setup from a thermal source. And as those of you who work with nonlinear light probably know, that you can get thermal light from, if you just take one arm of the output of an SPDC and ignore the other one. So if we throw away the heralds, we don't count them. I mean, they're still there, but we just don't count them, we ignore them. Then we get here effectively a thermal light distribution. We call this pseudo-thermal to avoid semantic arguments with people who say, no, it's not thermal because it doesn't come from sunlight. It's pseudo-thermal, but mathematically it's the same as a thermal distribution. And actually at the intensities that we're using, this is actually very serendipitous. The intensities we're using, the intensity of the light that's incident here is actually quite similar to that of sunlight. That's rather fortunate. Okay, so first of all, if we just look at the fluorescent light and we just measure the coincidence counts with single photon and then with thermal light incident, then we get these plots here. And I'm just going to first of all show that the fluorescence lifetime and the efficiency, which are quantities normally measured in photosynthesis, these are both independent of photon statistics. Basically, you extract the lifetime by fitting to this decay here, and the efficiency is also relatively easily measured. Okay, so that's really not so surprising. But what is interesting now is the second order coherence of the light. So we're looking at the second order coherence of the light, of the fluorescence, and of the incident light. And again, those of you who know some quantum optics will appreciate that second order coherence does tell you something about the quantum behavior of the light. It's very different between classical light and quantum light. And if there is some matter in between, which is giving rise to a change in photon statistics, then the implication is there's something interesting, likely quantum mechanical going on. There's something coherent going on, because it's affecting the coherent function of the light. So this experiment shown here, okay, so again, it's measuring these G2s under the different conditions. And what we see is, I showed you already the G2 of the incident photon, single photon incidence, and the fluorescence from that single photon incidence. So in this case, the input statistics, G2 is approximately 0. So here's a little table, the lookup table. If you measure the G2 at exact coincidence at tau equals 0, no time delay between the two photons, then if that function is greater than 1, you have classical light. If it's actually 2, then you have a single mode thermal light. That's actually hard to do. Normally one gets something close to 1. But if you have quantum light of the form of single photons, you will have something less than 0.5. And if you have pure single photons, you have 0. So that's a little lookup table if you're not familiar with this. So you see that with the single photon incidence, one has basically single photon emission. So the photon statistics are preserved. But if we have our pseudo-thermal light incident, so in other words, we're neglecting, we're not making any use of this herald. First of all, everything is much noisier in that case. So we're sensitive to all of the noise in the system, but we can make a very accurate measurement of the incident light G2 function. And this is, it's a little bit larger than 1, but only by a very small amount. And that's understandable because there's multiple temporal modes in the experiment because of the instrument response function. And then if we look at the fluorescence from this thermal light experiment, it's very noisy. It's really too noisy to fit. But we have here drawn, just to guide the eye a little bit, what would happen if you had ideal thermal light. And we see that we mostly have 1, but there's a lot of noise in this region around 0. It's not clear whether this is really meaningful. We need to do more experiments with higher time resolution. But what is clear from this is that the light is still thermal coming out. Okay, it hasn't gone completely changed to something that's less than, it doesn't have 0 here. So it's only the extent of the thermal light that has changed. So here's a comparison of these 2 experiments then. The fluorescence lifetime and quantum efficiency are the same. The fluorescence statistics and the statistics are essentially the same as those of the incident light, with the caveat about the change of the value of G2 from 1 to something possibly larger than 1 for the thermal, which I'll say a little bit more about in a moment. So the major conclusion is that light harvesting does preserve the statistics of the incident light. So what are the implications? So firstly, after a lot of thinking about it and doing some estimates and so on, we maintain that you would expect similar results for if you were using coherent light or laser incident light. Namely, you'd have the same lifetime and efficiency, and you would also preserve the coherent photon statistics. And there's a good reason for this. Will come up in the next thing. So then the immediate question is, what kind of internal dynamics could change the photon statistics? Is it possible that there could be a change in photon statistics? I mean, we know that interaction of light with matter can change the statistics of light. That's the whole principle of a laser. You pump thermally and you get coherent light out, but that's very, very high excitation rates. This is very low excitation rates. Are there any internal dynamics that could change the statistics? So one option is, so the obvious option from the perspective of light harvesting is this very widely, extensively studied vibronic energy transfer, which occurs on a timescale of picoseconds. However, our experiments are not sensitive to picosecond dynamics. That's coming up on the next slide. Our experiments really would only see, the experiment I showed you would only really be able to show to whatever was happening around 0, essentially would have to be on a nanosecond or a fraction of a nanosecond motion. And the only relevant dynamics of these systems that goes up to nanoscales is protein backbone vibrations. And there's no evidence so far that protein backbone vibrations are coherent. But this is the only possible explanation that we can think of that would account for an increase in G2 on the nanosecond scale. But let me now point out the limitations of the experiment so far. The real limitation for the pseudo-thermal experiments is the timing resolution, which is limited by the fluorescence detectors that we have. So we therefore we cannot have any information about energy transfer dynamics within or between complexes, which would be the vibronic energy transfer complex dynamics. So these are all picoseconds or shorter. Or also there's interesting options. There may be collective effects, there may be super radiant effects, and all of these things we'd have to go down to much, much shorter timescales to measure, to detect them. So what do you need? So you need this. you need to have some picosecond time resolution for the G2, and this could be done by a photon counting streak camera, and there's one on order, but it'll be a few months before we get to it. This is just an example from, this is Birzig and collaborators in Germany, that this is now a completely different setup. This is a solid state setup. They're looking at lasing from a micro pillar system, and so they see as they change the pumping rate here, and they're measuring G2 of tau on a picosecond timescale, and they can see as they increase the input power, they can then see the transition to a lasing setup right here. So that's where we want to go. So how much time do I have left?
I think we should wrap up soon.
Oh, okay. So I can finish basically. This slide is. I'll just maybe mention this slide, then I'll finish. This slide. So initially, when we started this, we said, okay, so after we've done these, the incident fluorescent correlation, then we'll go on and do pump probe with single photons. So it sounds great. Single pump photon, single pump probe. Here's a very simplistic scenario for this. And you have these advantages that one expects about from entangled light. Now we would be using the entanglement of the system of the light. But this is not experimentally feasible because of when you put the numbers in, you see that the probability for 2 photons to be absorbed by 1 complex in your sample, so whether you do it with a single molecule or not, you have to have 2 photons absorbed by the same complex. So that probability is very, very low. And it would probably result in a postdoc of 10 years rather than a postdoc to do the experiment, and it's just not feasible. This is another thing that we've considered, but this actually turns out it can be dequantized, and I don't have time for that. So let me just skip all the way to the end now and end with the conclusions. Yes, and the advertisement for a position. So we have a position available. Whaley is moving on to a faculty position, so we do have a new position for anyone who might be interested. If you know anyone who might be interested in carrying out the kinds of experiments that we're doing with quantum light. Going forward, we'll be looking at G2 measurements of single complexes and looking at, I think I missed something. One slide actually didn't go. You could change the, okay, so I missed one slide, but measuring G2 of fluorescence on a picosecond timescale would allow one to then probe the vibronic energy transfer processes in the light harvesting systems at the single photon level. And this would be very, very exciting for the field. That's what we want to do going forward now. Okay, thank you.
I guess we have time for 1 or 2 questions. Yeah.
Good work. Regarding the energy transfer followed by the fluorescence decay,
yes,
it takes some time. I wonder what kind of degrees of freedom are preserved and some are lost. I guess your temporal degree of freedom is retained, right? That's why you can do
in the experiment.
Yeah, the other degrees of freedom would be like polarization, spatial modes. In particular, for quantum imaging purposes, we're interested in the number of effective entangled modes or the Schmidt number.
Yeah. So we calculated the Schmidt number. It's in the supplementary material in terms of the frequency, entanglement, and so on. But polarization is a good point. Polarization is probably lost because these systems are disordered. But the next generation of experiments that we want to make we'd measure G2 with polarization dependence and also with frequency dependence. And if you do that, there's work by Alexandra Oleo Castro in London that shows that there would be things that you'd see not at 0, but you would see if you could measure G2 on a timescale of say of 10 or 20 picoseconds, you would see some interesting behavior that would reflect the internal dynamics. Yeah. And it would be sensitive to polarization.
I see.
And to frequency. So it's frequency selection. So of course all that makes the experiment harder because you're going to have to add frequency selectivity onto your detection as well.
By the way, is the source single mode or multi-mode?
The SVD source.
Your source for the integral photon pair source.
It's multi-mode in the sense that that's why we get G2 is close to 1.
I'm sorry, I mean the transfer spatial modes. Spatial modes.
Oh, the spatial modes is 1.
Is 1.
Yes.
Okay.
Yes, yeah.
Thank you.
Yeah.
Okay, maybe in the interest of time, let's take a break now and let's resume at 10:45.